Hi, my name is Kate Lathrop. I'm a breast medical oncologist at the University of Texas in San Antonio Mays Cancer Center, and welcome to our SABCS Snippet. I'm here with Dr. Erika Hamilton. She is the Director of Breast Cancer Research at the Sarah Cannon Research Institute. And we're excited to have you today, Dr. Hamilton, to talk to us a little bit about some exciting news from the HER2CLIMB-05 study that just had a press release.
Thanks so much for having me. Excited to discuss it.
Yeah, so just for the audience, can you just go over briefly the trial design and what you were specifically looking at here in this trial?
Yeah, so as we know, HER2CLIMB-05 was essentially a trial in the maintenance setting for first-line HER2 positive breast cancer. So right now, our standard of care has really been taxane in combination with trastuzumab and pertuzumab. And then after some amount of cycles, typically 6 or 8, although this can vary from person to person, the taxane is dropped out and the HP continues in a maintenance fashion. We often add endocrine therapy at that time as well. And so what HER2CLIMB-05 was testing is as that time point that the chemotherapy is dropped out, HER2CLIMB-05 allowed 4 to 8 cycles of chemotherapy, and we would be continuing just the trastuzumab, pertuzumab maintenance with or without endocrine therapy, the addition of tucatinib. We know tucatinib from the initial HER2CLIMB trial. It's FDA approved, HER2 specific tyrosine kinase inhibitor in combination with capecitabine and trastuzumab. And so this really was testing, looking at tucatinib in the earlier setting, first line maintenance. And then there are a few things we were particularly interested in around brain mets and delay to time to progression.
Yeah, so tell me a little bit about any of the results that you can divulge yet.
Yeah, so there was a press release that this trial, HER2CLIMB-05, did meet its primary endpoint. So the addition of tucatinib did delay progression-free survival in that first-line maintenance setting. We don't have a lot of the details available for public consumption yet, but this will be presented at San Antonio Breast Cancer Symposium in December. And so we're looking forward to diving into that data a little bit more at that time.
Yeah, we're certainly looking forward to hearing that presentation as well. It's exciting news. We've been using the same regimen in this setting for more than 10 years, the Cleopatra regimen. And in the last few years, we've seen this area get a lot more busy with a lot of other options. So I have a few questions for you there. One, can you talk a little bit about how you defined brain mets in the study, who was allowed on, if they could have prior brain mets, or how is that dealt with on the study?
Yeah, so again, this is a little bit of a unique study because it's really a maintenance study. And so patients weren't progressing at the time of entry. And so these patients could have had brain mets. But again, these patients were patients that had taxane, trastuzumab, pertuzumab, had good disease control, had received 4 to 8 cycles of chemotherapy. And at whatever time they or their physician was kind of planning on dropping out the taxane and their disease was under controlled, they would enter at this time. So a little bit different than your typical clinical trial where people are entering at a time of progression.
Okay, so you guys are looking at preventing the development of brain mets, not delaying recurrence, or both?
Both. So
Okay.
it's not recurrence. So this is first line metastatic. It's delaying time to progression.
Time
So
to
that
recur.
was the primary endpoint. That's what the press release was positive on. But key secondary endpoints were also around brain mets. We know that tucatinib has good brain activity in the initial HER2CLIMB trial. Almost 50% of patients actually did have brain mets at the time of entry, and it significantly improved PFS and survival among those patients that had brain mets. So I think one of the big questions in the field was taking a drug that really acts in the brain and has good activity and moving it up into the earlier lines. Certainly could we delay progression, et cetera, but also could we maybe prevent brain mets? And so I think that's one of the pieces of data people are looking forward to seeing.
Yeah, that'll definitely be exciting. So, um, how would you maybe discuss the way that this is going to fit in clinically with some of the other trials that have recently had results presented, specifically Destiny Breast 09 and also the Patina trial. Of course, the Patina trial is looking at, the hormone receptor positive setting alone, but where do you really see this maybe fitting in? And I know the data's early.
Yeah, absolutely. Those are great questions. So, we have seen data for patina fully. That trial was also in the maintenance setting, specifically among patients that had ER positive disease, similar design. Patients weren't progressing when they came on to the study, but it essentially randomized patients to doing their endocrine therapy with pertuzumab and trastuzumab, or adding palbociclib, one of our CDK4/6 inhibitors. And it actually showed, impressive improvement in progression-free survival from about 29 up to 44 months. That was, a very kind of subsetted population in terms of who was allowed on that trial. And you already highlighted, specifically only patients that had ER positive disease.
Hmm.
HER2CLIMB-05, a little bit different because patients were allowed whether they had ER positive disease or not, but also in that maintenance setting. And then the other trial that you referenced was DESTINY-Breast09. We haven't seen, everything about this trial. We saw this most recently at ASCO. This was looking at first-line upfront trastuzumab deruxtecan, so bringing trastuzumab deruxtecan up into the first-line setting. Really, again, profound improvement in progression-free survival, about a 13-month improvement. It's not FDA approved yet in
Right.
the first-line setting. And it raises a lot of questions because it doesn't have a maintenance strategy. There wasn't a part of that trial where the antibody drug conjugate was dropped out and patients might have gone on to their endocrine therapy or some type of maintenance that was a little bit less toxic. And so I think how all three of these are going to be integrated is a fantastic question. One we probably don't have all the answers to. I do think there's going to be a strategy for these maintenance regimens, whether we're talking about patina or whether we're talking about HER2CLIMB-05. Really progression-free survival in that first-line setting can be so long. And when we looked at trastuzumab deruxtecan, I mean, we're talking 3 years, 4 years. Some patients are probably 5 years. And to continue, a cytotoxic in that setting, isn't going to be as easy for patients to tolerate than dropping that out and kind of going to, antibodies or antibody with TKI or antibody with endocrine therapy. And so I think this is one of the zones that we're in a little bit of a data-free zone with DESTINY-Breast09. It's certainly a regimen that's going to be used, but I still think there's going to be a big appetite to think about maintenance strategies for these patients that have good responses to DB09.
Yeah, absolutely. And I'm sure you're going to be showing the data whether these patients were hormone receptor positive or not, and maybe see if we can further figure out maybe some can have the patina regimen and some would do better with this regimen. So it's exciting to have more options. Final question for you. How did the patients tolerate the tucatinib in this setting? Did you guys see any safety data you were concerned about? Can you comment on that?
Yeah, we haven't released a lot of the safety data. I can share with you that there was nothing surprising. So I think, what we would have expected from knowing tucatinib's side effect profile. And again, I think with a lot of these maintenance regimens, really dropping out that chemotherapy, patients feel so much better when the chemotherapy is gone and they're kind of on these maintenance regimens. So really compares quite favorably to cytotoxics.
Yeah. Well, Dr. Hamilton, thanks for taking some time out of your busy day today to go over these results with us. It's really exciting.
Absolutely. Thanks for having me.